All right. So we've been running this project for a couple of years, and we'll start with a little bit of introduction. So, I mean, I think you agree that the brain neuroscience field is in a different state than it was before. On the bottom right, we took a recent analysis that was done by Stevenson and Cording, where we showed the number of neurons that are recorded over time. And you might argue that lately we've been increasing pace, but there's a certainty here is that it's going up. And this is because of the new technology that are here on this slide. Multi-area calcium imaging that we're building here, multi-electrode probe recordings. And so this has posed the questions of how we operate as a field, because it's totally feasible that in the next 10 years we'll record 10,000, 50,000 of neurons. And so it sort of suggests the idea that maybe a model that has been used in astronomy is more feasible to scale, because it allows the different community, like the experimental, technological, and theoretical scientists, to work together in a marketplace of ideas. And so this idea has been here at the Institute for a long time. Christophe and Clay brought it here, but we thought it was time to test it. And so what is this about? So this is OpenScope. We sort of tried to test these ideas in the pipeline here. And so we will be interested in this. It's an open experimental platform to test model of cortical computation. And there are two paths to it. So computational scientists could form hypotheses based on analysis of our data and propose experimental collaboration with the Observatory. Or experimental scientists observe a new phenomenon in their own data set, and they want to validate it at scale and on our platform. In the last 2 or 3 years, we actually tested this idea, whether it's actually worked with our field, where we could have an observatory, and we're going to talk about this in this talk. And I'm going to hand it off to Josh, who is going to talk to you about the pipeline.
Hi, I'm Josh Larkin. I work in the optical physiology team. And I'm going to tell you a little bit about the infrastructure that OpenScope uses and how we operate. So we have been doing the Allen Brain Observatory for about 4 years. And you've heard about this already one time. And so basically, we breed the animals in-house. They go into surgery, and then they move through our pipeline. We have moved over the last 4 years into a state of such strong standardization that 5 weeks from the surgical date, we can count on the animals arriving in the 2-photon imaging state with a clear retinotopic map, easily handleable, and we can start data collection immediately. So this standardization is something that we see with the CAD model on the right, which you can find on 5 different microscopes upstairs, and they are all very, very standardized and collect high-quality data. But standardization itself is not enough to provide a service for the neuroscience community. We also need to be able to operate at scale. So here on the left is our surgical suite, which provides surgeries and animals flowing into our pipeline every week. And on the right, you can see for the original Brain Observatory paper, we had 422 mice move through this pipeline, of which 243 completed and were included in the analysis. So that is a large scale of learning to happen. And over those 4 years, we have managed to standardize and learn enough to avoid many of these modes of failure that are included in that figure. So how do we utilize this pipeline to dig into the scientific questions that our OpenScope partners have? One of the ways is adding retrograde labeling injections. We have had 2 projects that have done these injections in V1 so that we can look at specific neurons in the higher visual areas that project to V1. And one of the projects this year is doing these retrograde labeling injections in the higher visual areas so that we can look at the neurons that project to those HVAs. All 6 of our projects so far have changed the habituation paradigm, at least a little bit, one of which you'll hear more about later. Used habituation to create expectations in the mouse, and then once they got to the two-photon imaging stage, those expectations were violated. So we got to watch the neurons respond in that case. Some of the other ways that we've changed the traditional observatory pipeline is doing dendritic imaging or removing the stimulus screen entirely and adding a light bar that flashed 40 hertz light for the mouse. So all of this knowledge gives us a strong background in how our pipeline works and the feasibility that comes that we need to apply to the proposals, which Yazan will talk about.
Thank you, Josh. Hi, my name is Yazan Billeh, and I'm a scientist at the Allen Institute. And so I'm going to talk to you about how we went about choosing the projects that we felt would have the most scientific impact. And so the first thing that we had to do is actually inform people of this opportunity. And so we made a request for proposals. Every team then had to submit a letter of intent, and this was an application to tell us what they were thinking of doing, and we received applications from all over the world. We then would do a feasibility review. Is what was being asked possible? Can we also suggest maybe certain features that the applicant team didn't know about, such as a different query line or mouse line that maybe could improve things better? And we would give this feedback to the applicants. We really wanted to try and get the teams to come up with the best scientific proposal, and so we really tried to make the feedback as helpful as possible to really to be able to execute all of them if possible. We then get the full applications and the full proposals, and we would do a scientific and leadership review. And so for this icon here in particular, I chose a person who is wearing a blindfold intentionally, because both in the scientific review and in the feasibility review, we performed a double blinded review process. And the reason for that is we wanted to choose the best science. It didn't matter what stage of career you were, where you were from. We were going to choose it purely based on the science. And there was actually a nice little result that came out from the Hubble telescope that is very encouraging along the lines we were thinking of. And so the Hubble telescope is in space, and people can apply for time on where to point it into the sky. And so this is a graph that is showing the number of PIs who were men and the number of PIs who were women across all the years. And the proportion of acceptance taking into account the number of proposals from both groups. And you can see that the blue, which represents men, is consistently higher year over year, just always until 2018, which is the first year they did a double-blinded review, and then it became equal or 50-50, more or less. So this tells us that now the Hubble telescope is choosing the best science with no inherent biases, and we were doing this right from the beginning. And after the scientific and leadership review is done, we actually select the proposals and inform the teams. We then had to execute the projects, and so we started this off with a kickoff meeting with the teams, and we determined what the teams will provide for us. Every team changed the stimulus, as Josh mentioned, and so we would need the scripts for the stimulus, but some teams also provided us hardware, where someone from the team came over to the institute and actually gave us new hardware that is needed for the experiments. We then generate the data here at the institute in this very building. This happens in a two-step process. First, we do some pilot experiments, and this allows us to work out all the things we need to be able to execute it in a seamless way, while at the same time it gives the teams an opportunity to test different hyperparameters of their models. Once we pass through the pilot experiments, the teams will do preliminary analysis, make sure everything's okay, before we go on to full production experiments and collection. Part of what is expected from every team is to write a written report, an analysis report for us, and then we're looking forward, and hopefully in the next couple of months, for publication and data release from all this data, from all the projects, or the first projects that were selected. And so these are sort of the selected projects for the first 2 years that have been selected. We have 6 in total, 3 every year, and the institutions are wide varying, and also the projects cover a wide spectrum, studying things like motion detection, but also studying Alzheimer's disease and treatments for Alzheimer's disease. using our platform. And you're going to hear about 2 projects now in particular from 2 different teams. And what's actually interesting here is their results actually show contradictory results. However, we can now be confident, because this is all happening in the same standardized pipeline, that this is not a methodology issue, and it's actually something with the biology. And so that's one of the advantages. And the first person to speak now is Joel.
Well, thanks. And as a former cosmologist who's used many of the shared telescope resources, now using the Brain Observatory, I'm just super excited to see neuroscience going in this direction. I think it's a real plus for the field. In particular, I'm a theoretical neuroscientist, and through the OpenScope, I was able to team up my lab with the labs of 3 other really wonderful theorists to test some ideas about how the brain might implement deep learning, or in other words, solve the credit assignment problem. This is sort of a cartoonish description of that credit assignment problem. The idea is that sensory information flows through multiple stages of processing before eventually turning into decisions or behavioral outputs. That means that any errors in those outputs could arise at any one of the stages of this processing, or in fact at multiple stages at the same time. And as a result, if the animal is going to learn to do a better job, in other words, to reduce those errors, they somehow need to figure out which stages of the chain are to blame for any errors in the final output. That's known as the credit assignment problem, and it's just that. How does each neuron in this chain know its contribution to system-wide errors? In the machine learning field, of course, we've engineered solutions to that problem. The best known is backpropagation or backprop, and using backprop or target prop and related algorithms, people can train deep neural nets like these to solve lots of interesting problems, right? Driving vehicles, identifying objects in pictures, and so on. The basic idea behind backprop is basically shown here. So this formula, delta W, gives the change in a synaptic strength, and it's a function of a few things. The first of these are this X is like the input to the synapse, and then the H is something like the output from the cell that the synapse is attached to. So both of these pieces of information are certainly available at synapses in the brain. The big question, though, is where this third part comes from, this neuron-specific error signal. And that's the part where it's not clear how every individual neuron has access to it. Now, in backprop, the way that delta, or neuron-specific error signal, is obtained is that a vector of error is computed at the output stage, and it's sent back through the connections in the circuit that would otherwise send information forward through the hierarchy. So it's back down through these Bs, and that results in this neuron-specific error signal. So we basically hypothesized that the brain might do something similar, and so we went out to search for these top-down communicated error signals that might be used in driving learning in the cortex. Now, in this particular case, the biology actually did us a favor by physically separating the top-down feedback signals from the bottom-up inputs to the neurons. Notably, pyramid cells in cortex, that's all the excitatory cells, about 80% of neurons in cortex, have this structure where they have this long apical dendrite that branches at the brain surface, and it's there in layer 1 of cortex that the pyramid cells receive their top-down inputs. And so the question to ask then is whether we see evidence of sort of error information present here in layer 1 of the cortex in mouse lines where the pyramid cells express the calcium indicator. The experimental setup that we devised is open loop, so there is not a behavioral task for the animals to do. That's one limitation of what we've done. However, we were able to exploit the fact that animals naturally have a predictive tendency, right? They want to know what's going to happen next so that they can behave appropriately. And so we set up the habituation period where for the week before the experiment, the animals experience this sort of stimulus, shown here cartoonishly, consists of bricks of light moving consistently in one direction. And it's the kind of stimulus you would see if you walked along an infinitely long brick wall, right? The whole world is consistently flowing in one direction. Then during the experiment, at random times, we break that expectation, shown here in sort of burgundy color, where a random subset of those bricks turn and start flowing counter to the rest of the world, thus violating the expectation of what should happen. And in the sense of unsupervised deep learning, that should cause an error in the brain's internal prediction that we should hopefully see communicated in a top-down fashion back to V1. while the mice experience these stimuli, we do our 2-photon imaging either at layer 1 of the cortex, in other words, to see those apical dendrites, or we put the focal plane down at the cell body layer, and we do this in 2 separate mouse lines, so we can either observe what the layer 2-3 pyramid cells are doing, that's shown here, or the layer 5 pyramid cells. And again, for both mouse lines, we see the apical dendrites and the cell bodies. These are what our data look like, or at least the sort of first analyses that I'm ready to share. So the layer 2-3 pyramid cells are shown here, layer 5 pyramid cells in this other column. The first session is sort of the first time the mice ever see this violation of expectations, and then on another day is session 2, session 3. Dendrites are up here, cell bodies are down here. A couple things to note. In layer 5 pyramid cells, there really is no evidence for error-related information. I should mention that sort of the expectation violations happen here where this dashed line is present in the plot, and these are aggregated plots over all of the ROIs and all of the animals that we have. So in layer 5 pyramid cells, no evidence of error-related information. In the layer 2-3 pyramid cells, both in the apical dendrites, so that top-down pathway, and at the cell bodies, we see strong error-related signals. And those are statistically significant in layer 2-3, but again, there's no signal in layer 5. So our sort of preliminary conclusions, if you will, from this are that we see top-down signals, in other words, apical dendrites being activated at times of errors in layer 2-3 pyramids, but not layer 5 pyramids. The latency actually changes over days in an interesting way. So if you look at the cell bodies, you'll notice the latency of the onset of these error events is about the same from day to day. So the delta time here and here and here is about the same. But if you look at the top-down pathway at the apical dendrites, that latency shrinks substantially, right? It goes down from sort of this amount down to being near instantaneous by the 3rd day. Importantly, that means that especially in the 3rd day that the animals experience these stimuli, the top-down feedback signaling of errors precedes the activation at the cell bodies, suggesting that the top-down error signal might be actually driving activity at the cell bodies. And that's been shown by work in Jeff McGee's lab to be a mechanism that can actually drive learning in pyramid cells, stimulating the apical dendrites to drive somatic activation. So tentatively, we see backprop-like error signals in primary visual cortex, although there's of course more analysis to do. Some of these are to look for changes in the way the somas respond to stimuli as a result of the top-down error signals, and compare what we see in our experiments to machine learning models where we can train the algorithms to do similar tasks. So I'll leave it with that and pass the mic on to Hannah.
Cool. Yeah, so in our OpenScope project, I feel extremely lucky to work with my awesome teammates, composed of Nick Kane, Marina Garrett, and Rylan Larson. So in our OpenScope project, we are also trying to look into error signals like Joel, but we are looking into the error signals more in the perspective of hierarchical predictive coding. So what is hierarchical predictive coding? So it's a theory of hierarchical coding computation, which postulates that the internal representation of the world is actively compared and updated via feedback signals, which carry predictions about the lower area activities, and feed-forward signals, which represents the residual errors between the feedback predictions and the encoding expectation. So it's been a very popular theory in neural coding and was popularized in the seminal paper by Rao and Ballard from 1999. However, so far, the detailed mapping between the cortical microcircuit and algorithmic nodes in predictive coding has not been very well studied, as pointed out by this review paper came out last year by Keller and Merzik-Flogel. So in our OpenScope project, what our team is trying to find out is to tackle this question: does predictive coding exist in mouse visual cortex? All right, so here we came up with 2 main aims. So first, are there prediction and error signals across 3 hierarchically connected areas, namely primary visual cortex, VSPM, and RSP in mouse visual cortex? And our hypothesis is yes, and we expected to see more error signals in the primary visual cortex because it's lower sensory areas, and more of the prediction signals in the higher cortical area like RSP. And if so, are prediction errors more prevalent in specific cortical layers? So there are actually two candidate layers for encoding prediction errors. One is layer 2, 3, and the other one is layer 5 and 6, because both layers receive the bottom of sensory inputs as well as the top-down feedback signals from the higher cortical area. And our hypothesis is that error signals are computed in layer 2 and 3, and predictions are encoded in layer 5 activities. To prove this, we implemented two different types of expectation violations in our experiments. So first, we introduced temporal expectation by using learned sequence violation, where we present the animal with the sequence ABCD of natural images, so the mouse is highly habituated, and after the repeats, the last image here, the D, is replaced with an audible image, and then also the audible. So the mouse is also very heavily habituated with the audible images as well. So it's not just some novelty effect, but it's actually about the sequence violation. Okay, we also included a second type of expectation violation, which we call spatial expectation violation, by introducing different degrees of occlusion on the familiar images. So, like, can be 0%, 15%, 30%, 40%, all the way up to 60% occlusion. All right. So here is what we found from looking into the sequence violation signals. So in order to quantify the sequence violation signals, we introduced something called what we call audible response index, which is a normalized difference between the response to audible images during the sequence violation and response to image during the transition control. And when we looked at the distributions of this audible response index, this is what we see. So we actually saw that the sequence violation signal distributions are actually pretty much similar across all different areas, VSP, VSP, and RSP, and also across different layers at different depths. However, the sequence violation signals are not just some generic audible signals, because sequence violation signals are image specific, as we can see here from this distributed light bands. Okay. When we looked into the occlusion population response, we also saw that the occlusion signals are also everywhere in different areas, as we see here in the distribution of the occlusion signals across three different regions, and also across different layers. However, when we looked into the sensitivity of the occlusion signals, they were actually different across the regions and the layers. So, so what we meant by the sensitivity here is that we can actually look at the response of the occlusion response to different degrees of occlusion. And as we see here, in the earlier areas like VSPM, the response actually decreases with added occlusion. However, that trend changes in the higher cortical area, like in RSP. All right, so here are our initial findings. So first, we actually found that the prediction error signals for both temporal and spatial expectation violations are present in all three areas across all layers, which is in disagreement with the previous expectation from the previous studies, where we assumed that the prediction error encoding is happening in some specific cortical layers. However, we found that the sequence violation signals are image-specific rather than being some generic audible responses, and sensitivity of the prediction error signals encoding partial occlusion changes across regions and layers. So as for the next steps, we are working on mapping area encoding neurons to feed-forward versus feedback projection neurons based on injection location. We also want to investigate behavioral correlates, for example, by looking into the pupil diameter changes versus prediction violations. And finally, we'd like to classify neurons which respond both strongly to both temporal and spatial expectation violations.
So to conclude, I hope we've shown you that Observatory is open a new path to experimental knowledge. And to do so, it does create a marketplace of ideas. You already see that some results contradict one another, and so there's more work there to sort of merge these studies together. And some of the future technical capability of OpenScope we're really excited about are behavior tasks that we already have running in the pipeline, as well as multi-area electrophysiology and multi-area imaging. That said, I want to make sure to thank some founders, Paul Allen for his vision and support, as well as all the team behind collecting the data, all the arrays across all the pipelines that are working really hard to make this working really well. Thank you.
Questions for the OpenScope team?
Hi. My question is more for Joel. So recent work from Burkhalter lab has shown that the dendrites of layer 2-3 receive input from midbrain locomotor region. So for that reason, I guess it's especially important to show that your responses are not confounded.
Yeah, that's a great point. So we have the running speed of the animals, and we have the eyes tracked. We've extracted the pupil diameter. We did not see any correlations in our data when we looked, so correlations between running speed and layer 1, so apical dendrite activation, but that is a key confound.
I think one of the interesting things about the predictive coding projects and theories is that in backprop, to sort of like allude to the way that Joel set it up, you can't just get away with positive error encoding. You also need negative error encoding. And I wonder if either of you have built into your projects a way to look for that and what you think it might look like, whether you've seen anything like that.
Yeah, so you can easily signal both positives and negatives with all positive numbers by looking at deviations from a baseline, right? So the idea is if you have sort of generically some amount of feedback and you have more than that generic amount, that's a positive signal. If you have less than that, that's a negative. All of the data I showed was baseline subtracted, but we should ask whether that baseline is non-zero, and as a result, whether there's sort of the capacity in the top-down feedback to carry both positive and negative error information.
Hi, I was curious if you presented these somewhat contradictory results, but do you have the capacity to speculate a little bit as to what could be the differences? The differences in stimuli are that some expectations are purely temporal or purely spatial or a combination of that. Some of them are restricted spatially. Are there going to be any theoretical concepts which would be possible with both of the results you've seen?
Well, so I think one can be a can be the stimulus, but at the same time, what Joel looked into is he looked into VSP only, and he looked into two different types of cells, while we basically just looked into all different cells across all different layers. So that may be. So I don't think it's entirely contradictory.
Eric also has a question.
This is a question for, I guess, mostly Hannah and Joel. Having gone through the process once now and collected data and starting to analyze it, could you see value in another iteration, right? So applying again, maybe to do a follow-up study as part of OpenScope.
Yeah, I want to do a supervised task, so a behavioral experiment where the animal can make errors and use the multiplane imaging to look simultaneously at apical dendrites and cell bodies in multiple brain areas, with the idea of actually being able to see the flow of information at error times from deeper areas back to VisP. So yes is the short answer.
Yeah, definitely yes, because otherwise I would never be able to like participate in the experimental study as a theorist. Also, yeah, I think one of the original aim was actually looking into different cell types as well, using different transgenic Cre lines of mice, which was not really feasible during like when we were actually carrying out this project. So yeah, that may be the next follow-up study.
I wanted to ask quickly about credit assignment to both of you, both Hannah and Joel. Joel, you were looking for the error signal itself. Hannah, you were looking for violations from expectations. This B, I always thought, this neuron-specific code for how that error enters and should update a specific synapse. I always thought that that was like the crucial non-local component of the signal. And I know, Joel, some of your collaborators have advances in other directions. Maybe you can just tell us, though, as a large group, what do we need to see in order to solve the credit assignment problem, this big whammy from neuroscience, and at what level of neuron specificity?
Yeah, so the matrices that carry the top-down feedback information, sort of top-down connection weights, in vanilla backprop, those are the transpose of the feed-forward weights, which creates this non-locality Eric's referring to called the weight symmetry problem. Tim has a nice paper. The title is something like Random Feedback Alignment, basically showing that the top-down feedback connections can be random, so there does not need to be any structure there. So based on the theory world, the sort of basic components that are needed are just for there to be some vectors of error sent down through some top-down feedback matrices. We see in our data something that is sort of satisfying a necessary condition for that to be there, not necessarily a sufficient condition. But I would say that if we can demonstrate in our analyses that those error signals drive learning in the V1 neurons that are targeted, I think we'll be pretty far along the path to convincing ourselves that we are seeing credit assignment in action.
And also, I think the biology is there, and then we are basically like using different theories. So Joel is actually looking into backprop error. So like the error should be a feedback thing. So it actually comes down from the higher areas, and then like from predictive coding theory, what we are looking into is this like feed-forward error signals. But at the same time, maybe it's all like distributed all across, and then I think. Yeah, I think more like precise experiments and more precise study should be done before we can actually claim anything.
All right, thank you, everybody, again.